Assalamu alaikum. Welcome to my channel Easy Way. And in this topic, we will be able to know about what actually is polymerase chain reaction and why we are using it. So, polymerase chain reaction is the most important topic of this chapter, and this is the basis of the biogenetics, biotechnology. It is actually the basis of the biotechnology. So, first of all, we will know what actually is PCR. PCR is the polymerase chain reaction. This actually is named by the enzyme polymerase, due to which these reactions are proceeding. So, and this technique was developed by K.D. Mullis in 1983, and he was also awarded Nobel Prize in his significant research and the invention. So, PCR, what actually happens in a PCR, we take a gene or a segment of a DNA and actually it is amplified. Amplification doesn't mean that it is make much larger and bigger, but actually in number. Amplification means in number. Like if there is only one copy, multiple copies, like 4 to 5 copies are created by this polymerase chain reaction, and it is done again and again. So that is why this is the chain reaction. Repeated cycles are there. So very specific, it is very specific. Like if you want to done the gene cloning of a very short segment, bring those primers to that sequence, and the DNA polymerase will um, make the copies, and it will be and amplified, like multiple copies will be um, done. So the polymerase chain reaction is like this way, and as you can see in the diagram to 23.5, if take any DNA template that is from 3 dash to 5 dash, and then we will heat it, so that we will heat it for its denaturation. Why there is denaturation? To break the hydrogen bonds between the two strands. When the two strands are separated, then they are cooled for 2 minutes, and then the primers are added, and this process is known as annealing. After the addition of primers, DNA polymerase will bind itself uh, with the primer, and the um, copy of that DNA segment or the gene is made, and you can repeat the cycle again and again, and in this way the specific number of that gene is increased. So before carrying out the PCR primers, as I have told you in that in figure, we have to add primers before the polymerase, because polymerase enzyme does not start a reaction, but only it continue it. So to start a reaction you have to add primers, and then polymerase, as the temperature in the PCR machine is up to 90 degree. So the human enzyme DNA polymerase does not work in 90. Its optimum temperature is 37 degree, 37. So we will not use the DNA polymerase of a human. We will use a DNA polymerase of a bacterium, Thermus aquaticus, and that polymerase is known as Taq polymerase, and it can be used in PCR. So PCR is and that can easily repeat the process again, and it is heat sensitive. So PCR machine is a thermocycler, and it is a routine equipment in any laboratory, and it is used again and again. So this was all about PCR. Any questions regarding this topic can be asked, and I will answer all those questions. Thank you.